Good day, this is Professor Will, CBMD, PhD. Today is Friday, December 21st, 2018. It is 18:34 seconds Eastern Standard Time. Science News. 300 blind mice uncovered genetic causes of eye disease. Date, December 21st, 2018. Source, University of California, Davis. Summary. Hundreds of new genes linked to blindness and other vision disorders have been identified in a screen of mouse strains. Many of these genes are likely important in human vision, and in the results could help identify new causes of hereditary blindness in patients. The results are the latest to come from the International Mouse Phenotyping Consortium, of which UC Davis Mouse Biology Program is a founding member. The goal of the consortium is to identify a function for every gene in the mouse genome by creating lines of knockout mice that lack a single specific gene and screening them for their effects. Consortium researchers have previously identified a set of genes essential to life, genes linked to deafness and even those linked to hereditary bad breath. To date, the consortium has generated more than 7,000 strains of gene knockout mice, of which 4,364 have been characterized across 11 organ systems. Researchers and colleagues combed the consortium database for genes linked to eye and vision defects. They identified 347 genes, of which 86 were either well established as involved in eye disease or were associated with vision in some way. 3/4 of the genes, 261, were not previously known to cause eye disease in any species. In 2018, if someone has a form of hereditary blindness, we can identify the cause 50 to 75% of the time. In the remaining cases, we know the mutation is there, but we don't know where to look. Now, eye centers that do DNA sequencing can call back patients and screen them for these new genes. While the mouse and human genomes clearly differ, most human genes have analogous counterpart in mice. Scientists check the newly identified mouse genes against their human equivalents. The new genetic information can also enable new therapies for hereditary eye disease. In 2017, the FDA approved the first gene therapy for any disease, treating hereditary blindness caused by defect in the retinal gene RPE65. We expect that more and more of these genetic diseases will be treatable. Good day, this is Professor Will, CBMD, PhD. Today is Friday, December 21st, 2018. It is 18:42, 10 seconds Eastern Standard Time. Science News. A safe wearable soft sensor, biocompatible sensor, can be used in diagnostics, therapeutics, human computer interfaces, and virtual reality. Date, December 21st, 2018. Source, Harvard John A. Paulson School of Engineering and Applied Sciences. Summary, researchers have developed a soft, non-toxic wearable sensor that unobtrusively attaches to the hand and measures the force of grasp and the motion of the hand and fingers. We have developed a new type of conductive liquid that is no more dangerous than a small drop of salt water. It is 4 times more conductive than the previous biocompatible solutions, leading to cleaner, less noisy data. Harvard's Office of Technology Development has filed a portfolio of intellectual property relating to the architecture of novel soft sensors and is seeking commercialization opportunities for these technologies. The sensing solution is made from potassium iodide, which is a common dietary supplement, to glycerol, which is common food additive. After a short mixing period, the glycerol breaks the crystal structure of potassium iodide and forms potassium cations, K+, and iodide ions, making the liquid conductive. Because glycerol has a lower evaporation rate than water, and the potassium iodide is highly soluble, the liquid is both stable across a range of temperatures and humidity levels and highly conductive. Previous biocompatible soft sensors have been made using sodium chloride glycerol solutions, but these solutions have low conductivities which make the sensor data very noisy. And it also takes about 10 hours to prepare. We've shortened that down to about 20 minutes and get very clean data. The design of the sensors also takes the need of children into account. Rather than a bulky glove, the silicone rubber sensor sits on top of the finger and on the finger pad. We often see that children who are born early or who have been diagnosed with early developmental disorders have highly sensitive skin. By sticking to the top of the finger, this device gives accurate information while getting around the sensitivity of a child's hand. Scientists currently study motor function using the Motion Capture Lab SEAS and WYSS. While motion capture can tell a lot about movement, it cannot measure force, which is critical in diagnosing neuromotor and cognitive developmental disabilities. Early diagnosis is the name of the game when it comes to treating these developmental disabilities, and this wearable sensor can give us a lot of advantages not currently available. This paper only tests the device on adult hands. Next, the researchers plan to scale down the device and test it on hands of children. The ability to quantify complex human motions gives us an unprecedented diagnostic tool. The focus on development of motor skills in toddlers presents unique challenges for how to integrate many sensors into a small, lightweight, and unobtrusive wearable device. These new sensors solve these challenges, and if we can create wearable sensors for such a demanding task, we believe that this will also open up applications in diagnostics, therapeutics, human-computer interfaces, and virtual reality. Good day, this is Professor Will, CBMD, PhD. Today is Friday, December 21st, 2018. It is 18:57, 46 seconds, Eastern Standard Time, Science News. A mountain of evidence on air pollution's harms to children. Date, December 20th, 2018. Source, Columbia University Mailman School of Public Health. Summary. A new study organizes the available scientific evidence on the effects of air pollution on children's health. The article is the first comprehensive review of the associations between various fossil fuel combustion pollutants and multiple health effects on children, in the context of assessing the benefits of air pollution and climate change policies. The researchers say their goal is to expand the kinds of health outcomes used in the calculations of the health and economic benefits of implementing clean air and climate change policies, which are largely limited to the effects of air pollution and premature deaths and other outcomes in adults. The new paper aggregates research on outcomes including adverse birth outcomes, cognitive and behavioral problems, and asthma incidents. Policies to reduce fossil fuel emissions serve a dual purpose, both reducing air pollution and mitigating climate change with sizable combined health and economic benefits. However, because only a few adverse outcomes in children have been considered, policymakers and the public have not yet seen the extent of the potential benefits of clean air and climate change policies, particularly for children. The researchers reviewed 205 peer-reviewed studies published between January 1, 2000 and April 30, 2018, which provided information on the relationship between the concentration of exposures to air pollutants and health outcomes. The studies relate to fuel combustion and byproducts including toxic air pollutants such as particulate matter PM 2.5, polycyclic aromatic hydrocarbons PAH, and nitrogen dioxide NO2. A table provides information on the risk of health outcomes for exposure by study encompassing research on 6 continents. There is extensive evidence on the many harms of air pollution on children's health. Our papers present these findings in a convenient fashion to support clean air and climate change policies that protect children's health. The World Health Organization, WHO, has estimated that more than 40% of the burden of environmentally related disease and about 90% of the burden of climate change is borne by children under 5. Although that age group constitutes only 10% of the global population. The direct health impacts in children of air pollution from fossil fuel combustion include adverse birth outcomes, impairment of cognitive and behavioral development, respiratory illness, and potentially childhood cancer. As a major driver of climate change, combustion of fossil fuel is also directly and indirectly contributing to illness, injury, death, and impaired mental health in children through more frequent and severe heat events. Coastal and inland flooding, drought, forest fires, intense storms, the spread of infectious disease vectors, increased food insecurity, and greater social and political instability. These impacts are expected to worsen in the future. Good day, this is Professor Will, CBMD, PhD. Today is Friday, December 21st, 2018. It is 19:20, 26 seconds Eastern Standard Time, Science News. Genome offers clues to esophageal cancer disparity. Date, December 20th, 2018. Source, Michigan Medicine, University of Michigan. Summary, a change in genome of Caucasians could explain much higher rates of the most common type of esophageal cancer in this population. A new study finds it suggests a possible target for prevention and strategies, which preliminary work suggests could involve flavonoids derived from cranberries. We've known for a long time that esophageal adenocarcinoma primarily affects Caucasians and very rarely affects African Americans. We want to see if African Americans have something genetic that's protective. If we understand why people have low risk, that can lead to understanding how to prevent cancer in those at high risk. About 17,290 Americans will be diagnosed with esophageal cancer this year. Adenocarcinoma represents about 2/3 of the cases, but is rarely seen in African Americans. In a study published in Gastroenterology, researchers examined tissue with samples from African Americans and European Americans, including both those with esophageal adenocarcinoma and those without. They measured gene expression and protein levels and found a difference in the enzyme, called GSTT2. This was significantly higher in African Americans compared to Caucasians. Researchers found Caucasians have a duplication of a portion of the genome that appears to reduce the expression of GSTT2, This enzyme protects cell against oxidative damage, such as this type caused by reflux, a key risk factor for esophageal adenocarcinoma. They confirmed the initial findings by looking at expression and sequencing data from 1000 Genomes Project, an international initiative that used genomic sequencing to provide a comprehensive view of human genetic variation. This data reinforced that populations from Africa and African descent had non-duplicating genome, while all other populations around the world had the duplication. The risk factors for esophageal cancer, such as obesity and reflux, happen at the same time rate for African Americans and Caucasians. But African Americans are not getting cancer. We see the highest risk of cancer in people who have this genomic duplication plus obesity. It is not just the presence of duplication, but these other factors contributing to the damage. Rates of esophageal adenocarcinoma have increased 600% over the last 3 decades, caused by a rise of obesity and gastroesophageal reflux disease, or GERD. The other type of esophageal cancer, squamous cell carcinoma, is more frequently seen in African Americans. The researchers used cell lines in a rat model to recreate low levels of GSTT2. They saw more damage in these cells compared to those expressing high levels of GSTT2. Next, they used the cranberry proanthocyanidin extract and found it reduced levels of DNA damage in the esophagus of rats exposed to reflux. This suggests potential for preventing esophageal adenocarcinoma. The key with esophageal cancer is to prevent it. Many people don't know they have the disease until it is too late to treat it effectively. Researchers are considering a clinical trial to test using flavonoids derived from cranberries and chemo prevention agent. However, more research is needed to understand appropriate dose and potential side effects. Today, this is Professor Will, CBMD, PhD. Today is Friday, December 21st, 2018. It is 19:42, 9 seconds Eastern Standard Time. Science News. Micropores let oxygen and nutrients inside biofabricated tissues. Date December 20, 2018. Source Penn State. Summary: micropores in fabricated tissue such as bone and cartilage allow nutrient and oxygen diffusion into a core, and this novel approach may eventually allow lab-grown tissue to contain blood vessels, according to the researchers. One of the problems with fabrication of tissue is that we can't make them large in size. Cells die if nutrients and oxygen can't get inside. Inside cells also do not differentiate if the chemical cocktail that triggers stem cells do differentiate does not reach them, a porous structure that allows both nutrients and other fluids to circulate. The researchers are trying a novel approach in creating tissue building blocks with micropores. They consider this an alternative to vascularization, growing blood vessels in the tissue, and call the outcome porous tissue strands. The researchers are starting with stem cell derived from human fat and mixing them with sodium alginate pyrogens. Derived from seaweed, sodium alginate can be printed into tiny particles that, when dissolved, leave behind tiny holes, pores, in the fabric of the tissues. The team uses the mixture to 3D print strands of undifferentiated tissue. They can then combine the strands to form patches of tissue. When the researchers expose the tissue to the chemical cocktail, it turns the stem cells into specific cells, in this case bone or cartilage. Because of the pores, the fluid can flow to all the stem cells. Researchers report in the recent issue of Biofabrication that the strands maintain 25% porosity and have pore connectivity of 85% for at least 3 weeks. By 3D printing strands next to atop each other as shown in their previous work, the strands self-assemble to form patches of tissue. These patches can be implanted in bone or cartilage, depending on which cells they are. They can be used for osteoarthritis, patches for plastic surgery, such as the cartilage in the nasal septum, knee restoration, and other bone or cartilage defects. In some ways, cartilage is easier than the bone, because in the human body, cartilage does not have blood vessels running through it. However, some bone is naturally porous, and so porosity is valuable in replacing or repairing that bone, while currently only tiny patches can be made. These patches are easier to fabricate than growing artificial tissue on scaffolding. The researchers are considering applying the same methods to muscle, fat, and various other tissues. Good day, this is Professor Will, CBMD, PhD. Today is Friday, December 21st, 2018. It's 20:01, 31 seconds Eastern Standard Time. Science News. Suboptimal inconsistent treatment for anaphylaxis due to unknown cause. Date December 20, 2018. Source McGill University Health Center. Summary: a new study is shielding light on anaphylaxis due to an unknown trigger, AUT, an unpredictable and potentially fatal allergic reaction about which surprisingly little is known. In practice, followed nearly 4,000 cases of anaphylaxis seen in Canadian emergency departments from 2011 to 2018. Common causes of anaphylaxis include food, medication, insect stings, latex, and exercise, but in some episodes no definite cause can be determined. With food, anaphylaxis avoidance of the triggering food is a key principle of management, but with AUT possibility of sudden life-threatening anaphylactic episode could occur at any time. In our study, 7.5% of the anaphylaxis cases were due to an unknown cause, and we found that the treatment and follow-up of AUT were suboptimal and inconsistent both inside and outside of the hospital. Specifically, epinephrine was underused, and antihistamines were the primary treatment drug for AUT. 46% of patients in our study received antihistamines before arriving at the hospital, and 55% of patients received antihistamines in the hospital emergency department. This research is the first longitudinal large-scale study to assess the clinical characteristics, treatment, and follow-up management of AUT cases across Canada. The study is part of a larger national project and database called the Cross Canada Anaphylaxis Registry, CCARE, which tracks the rates, triggers, and management of anaphylaxis in different provinces and settings across Canada. Among the study other findings, nearly one-fifth of patients with AUT were not prescribed an epinephrine autoinjector. Adults were less likely to receive an epinephrine autoinjector prescription than children. Only 56% of patients with AUT were referred to an allergist. Most of these were children. Of the patients assessed by an allergist, a trigger for their reaction was identified in 38% of cases. The underuse of epinephrine and the low rates of referral to an allergist, mainly in adults with AUT, highlight the need for clear guidelines and educational programs for diagnosis and management. The important takeaway from this study is that there are significant discrepancies in treatment and follow-up of patients with AUT that must be addressed in order to ensure prompt and appropriate management of anaphylaxis. Today's is Professor Will, CBMD, PhD. Today is Friday, December 21st, 2018. It's 20:31, 43 seconds Eastern Standard Time. Science News. How dietary fiber and gut bacteria protect the cardiovascular system. Date December 21st, 2018. Source: Max Delbruck, Center for Molecular Medicine and Humboldt's Association. Summary: this fatty acid propionate helps defend against the effects of high blood pressure, including atherosclerosis, heart tissue remodeling. A study of mice has found gut bacteria produce a substance which calms the immune system cells that drive up blood pressure from natural dietary fiber. You are what you eat, as the proverb goes. But to a large extent, our well-being also depends on what bacterial guests in our digestive tract consume. That's because gut flora help the human body to utilize food and produce essential micronutrients, including vitamins. Beneficial gut microbes can produce metabolites from dietary fiber, including a fatty acid called propionate. This substance protects against the harmful consequences of high blood pressure. Researchers fed propionate to mice with elevated blood pressure afterwards. The animals had less pronounced damage to the heart or abnormal enlargement of the organ, making them less susceptible to cardiac arrhythmia. Propionate works against a range of impairments in cardiovascular function caused by high blood pressure. This may be a promising treatment option, particularly for patients who have too little of this fatty acid. Detour via the immune system. Our study made it clear that the substance takes a detour via the immune system and thus affects the heart and blood vessels. In particular, T helper cells, which enhance inflammatory processes and contribute to high blood pressure, were calmed. This has a direct effect on the functional ability of the heart. For example, the research team triggered heart arrhythmia in 70% of the untreated mice through targeted electrical stimuli. However, only one-fifth of the animals treated with the fatty acid were susceptible to an irregular heartbeat. Further investigation with ultrasound tissue sections and a single-cell analysis showed that proportion also reduced blood pressure-related damage to the animal's cardiovascular system, significantly increasing their survival rate. But when researchers deactivated a certain T-cell subtype in mice's bodies, known as regulatory T-cells, the positive effects of propionate disappeared. The immune cells are therefore indispensable for the substance's beneficial effect. Short-chain fatty acid, a therapeutical option. The results explain why a diet rich in fiber, which has been recommended by nutrition organizations for many years, helps prevent cardiovascular diseases. Whole grain products and fruits, for example, contain cellulose and insulin fibers, from which gut bacteria produce the beneficial molecules like propionate, a short-chain fatty acid with a backbone of just 3 carbon atoms. Previously, it had not been clear which fatty acid is behind the positive effects of how it works. The study opens up new avenues in the treatment of cardiovascular diseases. It might make sense to administer propionate or a chemical precursor directly as a drug. For example, when the blood of those affected contains too little of the substance. Propionate still has to prove itself in everyday clinical practice. The research team now hopes to validate their findings by examining the substance's effects on human subjects. It is already known that propionate is safe for human consumption and can also be produced economically. The substance has been used for centuries as a preservative. For example, it is already approved as a food additive. With these favorable conditions, hopefully propionate will soon make the leap from the lab to patients who need it. Today's is Professor Will, CBMD, PhD. Today's Friday, December 21st, 2018 is 20:51, 19 seconds Eastern Standard Time. Science News. Statins are the most effective for those who follow the Mediterranean diet. Date, December 21st, 2018. Source, Neurological Mediterranean, IRCCS. Summary. For those who have already had a heart attack or a stroke, the combination of statins and Mediterranean diet appears to be the most effective choice to reduce the risk of mortality, especially from cardiovascular causes. The traditional Mediterranean diet is rich in fruit, vegetables, legumes, cereals, olive oil, wine in moderation, fish and low in meat and dairy products. Research says that statins and Mediterranean diet together were more effective as compared to one or the other considered separately in reducing the risk of cardiovascular mortality. Likely a Mediterranean diet facilitated the beneficial effect of statins that in our real-life study were generally used at low doses. Researchers also analyzed the potential underlying mechanisms of this positive interaction so far poorly explored between drugs and eating habits. The favorable combination of statins and Mediterranean diet appeared to act rather than on cholesterol levels by reducing subclinical inflammation, a condition that predisposes to a higher risk of illness and mortality. This finding is of a particular interest, especially in the light of our observation that a high level of subclinical inflammation doubled the risk of mortality in patients who already had a heart attack or stroke. Research suggests that we should focus more on the possible interactions between food and drugs, an aspect largely neglected in epidemiological research. Of course, controlled clinical trials will be needed to clarify these findings. If our data will be confined, new therapeutical possibilities could be designed for those who have already had a cardiovascular event, allowing a better modulation of the pharmacological intervention in relation to life habits. This is a new aspect of personalized medicine. Today, this is Professor Will, CBMD, PhD. Today is Friday, December 21st, 2018 is 21:03, 6 seconds Eastern Standard Time. Science News. Diabetes drug could be used to treat common heart failure syndrome. Date, December 19, 2018. Source, Rockefeller University Press. Summary. Researchers have discovered that metformin, a drug commonly used to treat type 2 diabetes, might also be used to treat a common heart failure syndrome that is predicted to affect over 8% of people ages 65 or older by the year 2020. The study shows that metformin relaxes a key heart muscle protein called titin, allowing the heart to properly fill with blood before pumping it around the body. Nearly half of the heart failure patients are considered to have HFpEF, in which the heart can properly contract. But because the wall of the left ventricle is stiffer than normal, it fails to fully relax between beats, reducing its capacity to fill with blood. This reduces blood supply to the rest of the body, leading to shortness of breath with exertion and difficulty exercising. HFpEF is more common in women, and other risk factors include hypertension, old age, obesity. Unlike other forms of heart failure, however, there are currently no drugs available to treat HFpEF. Researchers decided to investigate whether metformin could be an effective treatment for HFpEF because the drug has been shown to increase left ventricular dilation and lower the rate of heart failure in diabetes patients. Researchers gave metformin to mice with HFpEF-like symptoms and found that the drug reduced the left ventricular stiffness, thereby improving the animal's capacity for exercise. The researchers determined that metformin relaxes the left ventricle by making a heart muscle protein called titin more compliant. Titin acts like a molecular spring that helps the muscle recoil after it's stretched, and titin stiffness can be tweaked to enzymes that add phosphate groups to the protein spring-like elements. One of these elements, known as the N2B element, contains abnormally few phosphate groups in the HFpEF patients, making titin extra stiff. Researchers and colleagues found that metformin treatment increased the number of phosphate groups in the N2B element of mouse titin, causing the protein in the heart muscle as a whole to become more compliant. We therefore conclude that metformin is a potential therapy for patients with HFpEF because the drug is already approved and well tolerated in humans. Using it to target titin stiffness presents a unique opportunity for immediate translation to the clinic.